Dina, oh Dina, where are you? DNA structure and replication by Miss Hoskins. All right, so for DNA structure, you're going to learn how to analyze genetic information, how it's expressed in cells, and then we want you to be able to explain how the structure of DNA and even RNA are going to lead to the expression of information in the cell, and that is through a process of replication, transcription, and translation. When a cell is ready to divide, it must first copy its DNA. That process of making an identical copy of DNA is called replication. And that's DNA makes a copy of DNA, replication. This happens in the nucleus. And remember, it's during the S phase of the cell cycle. So there's a point when one of the cells has double the amount of DNA that it needs. Why? Because it's getting ready to divide. And so that S phase, synthesis phase, that's all part of interphase, is when replication happens. DNA replication ensures that each new cell will have exactly the same DNA as the cell that it's making. This enzyme called helicase will run down the strand of DNA, unzipping it, and that DNA will come apart into two separate strands. The openings are called the origins of replication, and remember, they're going to be anti-parallel. If we're starting in this direction, one origin is going to be in the 3' direction and the other one's going to be in the 5' direction. Several places along the DNA will be unzipped at once. So it's not like the zipper on your jacket where, you're unzipping one zipper at a time, it starts to come apart. Imagine having like 10 zippers on your jacket and you're unzipping it at one time, and then the zipper that is above it is closing the unzipped component. So this is all running, and it's easier to see in an animation here. But anyway, where it's starting is helicase is going to come and unzip the DNA and start the replication process. Enzyme DNA polymerase will add the complementary nucleotides to the template strand, the original strand that's there. And so as it works in the 5' direction this way, DNA polymerase, which is an enzyme, is going to pop in the complementary base pair that should be there. So DNA polymerase will add a thymine next to an adenine, and add an adenine next to a thymine, and then a thymine next to an adenine, and I'll add a guanine where the original strand over here has a cytosine, add a guanine to a cytosine, and it's going to do that in the opposite direction of the other strand. So DNA polymerase is heading this way on this strand, adding a thymine to the adenine, a cyanine to the guanine, an adenine to the thymine, and so on. DNA replication, part 2. It only works with the help of enzymes, all right? Primase is an enzyme that synthesizes short RNA sequences called primers. So it's required for DNA synthesis, and it's like a key. It kind of like starts the ignition process of replication. It makes short RNA primers. Those are like little short pieces of RNA to kind of help get the DNA polymerase started. DNA polymerase adds nucleotides to that RNA primer, and it starts to make polynucleotides. After all the nucleotides are added to that complementary strand of RNA primer, it's removed, and then it's replaced by DNA, and actually by DNA polymerase. DNA polymerase is then going to proofread the strand before the backbone is all finished up. And then DNA ligase comes by and it seals the gaps in the DNA. It kind of connects the DNA pieces by making this phosphodiester bonds. So here we have a piece of DNA replication in action, and you can think of this being an S phase where the DNA is copying itself. And if you look right here, we have some familiar enzymes. Here is DNA polymerase. It's also down here. Here is our helicase, the star of this show. He is unzipping the DNA strand. We also can see that the strands are anti-parallel. 3' is running this way, 5' is running this way, and as it is unzipping in this direction, we can see that RNA primer right here is replacing pieces, and along the side over here we have ligase that is moving up and down the backbone, putting the backbone in and repairing necessary bonds. After DNA replication has taken place, there are two identical DNA molecules formed. Each of them has like an old strand and a new strand. We call this semi-conservative replication. Semi-conservative replication, each parent strand is now a template or a pattern that determines the order of the new bases and how they're put on. And it's forming this complementary strand to the original strand. And the newly synthesized double helix is a combination of the old, or original, and the new DNA strand. As you can see from the diagram, from the old parent strand, part of it has gone to each new strand. We have formed these guys, which are complementary to the original strand. DNA replication, unzipping the DNA, enzymes help find complementary bases and bind them according to that base pairing rule. If you remember, adenine bonds with thymine and cytosine bonds with guanine. And those two identical DNA molecules are formed, each of them with an old strand and a new strand. So if you're thinking, consider like what kinds of mistakes could occur during this process. It's a very intricate, replication process. What could those implications be of those mistakes that could happen? What would be the difference in a mistake in the DNA of our bodies, like a brain cell, versus the mistake in a germ cell that would eventually become a sperm cell that we would, give to our offspring? Would there be a difference in the mistake if a mistake was made? And what would be the difference in a mistake in a gene that happened to a piece of that DNA versus to an overall chromosome error altogether? What are the outcomes of this? A huge chromosome. Is there a difference in the mistake that would make versus a gene, a piece of that chromosome? Or what if there's just one mistake in a letter?